Okay, welcome back, and here we are at the end of chapter 4. And what we're going to look at here is just a little bit of review on virology. If you've covered virology, this will be purely review. If you've not discussed any virology before, there's maybe some new stuff. And then we're going to look at viroids, which are weird and very cool biologically speaking. And then we'll finish out with something which you've probably seen in the press over recent years, prion biology. So let's get into viruses real quickly. There are several types of virus, and viruses are often kind of grouped together in the field of study with the viroids and the prions, and these are all kind of acellular infectious agents. And there are some viruses which cause diseases in animals and plants and infect bacteria too. Viroids are limited to plants and cause disease in plants, and then prions, as far as I know, restricted to animal infections. So let's start with the viruses here. These are small, very small acellular particles which can infect animals, plants, fungi, bacteria. If there is a biological entity, it's probably being infected by a virus at some point. So they are completely incapable of free living existence. In that sense, they are acellular parasites. They have no cytoplasm, no organelles, no ribosomes, and they only consist of a few types of protein and usually an RNA or DNA genome. So viruses are usually segregated into RNA viruses and then DNA viruses. So what viruses basically do is they infect a host cell, then they take over the host cell's replication machinery, and they then replicate more of themselves. Then they're released from the host cell either by budding or by causing the host cell to burst open. In terms of their naming, they're often named after the disease they cause. Poliovirus, causing polio in humans, tobacco mosaic virus causing infectious disease in plants, and then you will be familiar with the human immunodeficiency virus, HIV. So a lot of these viruses are named after the diseases they cause, but not all viruses cause disease, and so there are alternative naming conventions as well. Viruses are very, very small. Smallest is about the size of a ribosome, and then the largest are about the quarter of the length of a bacterium, so maybe 500 nanometers in length. There are some giant viruses, the so-called mini-virus, which have been recently identified, and these are cool and they some effects look more like cells than they do viruses, but they're classified as viruses. So like we said, they're chemically pretty simple. Going to have an outer shell, an outer coat of protein called the capsid, and that's going to surround the nucleic acid, and those are called naked viruses. Some viruses have a lipid bilayer which is derived from the cell they infect, and those are called enveloped viruses. So HIV is an enveloped virus. The herpes viruses, which is a big family of viruses, those are also enveloped viruses. Pretty simple. However, the genetics and the replication of the genome can be incredibly complicated. So that's all we're going to get into with viruses, and they'll come up probably as a tool used to study cell biology a little bit later in the semester. Viroids, these are even simpler. How can you get more simple than a virus? Well, you get a viroid. So these are infectious agents found in some plants, and they're most problematic in crop plants. They are small circular pieces of RNA. That is it. That is what a viroid is. Just a small piece of circular RNA. And you can see one here. This is potato spindle tuber viroid, and you can see it looks like DNA because it looks like it's double-stranded, but if you looked at this really closely and the resolution was good enough, you'd see it contains uracil. And so it's a closed circle of RNA with regions of complementarity, and so you can see you get these double-stranded regions along the length of the molecule. They cause diseases in several important crop species. You may have heard of kadang, which is a problem with coconut palm. So if you use palm sugar, then that affects coconut palm. Here you can see a potato spindle tuber viroid. On the left, these are normal potatoes, and these are potatoes that have been infected by the viroid. And so you can see the potatoes are small, they're stunted, their growth is completely abnormal. And so if you're looking for potatoes this big and you get potatoes this big, that's obviously an impact on people using these crops. Like viruses, the viroids don't exist freely. They have to be replicated inside the cell cytoplasm, and they are transferred from plant to plant by direct contact. So if there's a surface of a damaged leaf or something brushes against the surface of another damaged leaf, that's how they're transmitted. So they don't get airborne, they can't spread on animals or anything like that. They are transmitted purely by the direct contact between damaged surfaces of plants. Okay, let's finish up with prions. Moving away from nucleic acids there for viroids, or RNA for viroids. Prions are proteinaceous infectious particles. They're responsible for a number of neurological diseases. You've probably heard of scrapie, which is where sheep get a prion disease in the brain, and it causes them to rub up and down on fences and gates and trees and that kind of thing, hence the term scrapie. There's a disease of humans called kuru, which was once endemic in the tribal populations of Papua New Guinea, and now I believe it's been eradicated, and that was due to a ritual ceremony where the brains of elders were eaten by the young in the tribes, and that led to neurodegenerative disease in those populations. You've probably heard about mad cow disease. Certain people in the United States, myself included, cannot donate blood because of the mad cow outbreak in Europe many years ago, maybe before some of you guys were born. But mad cow disease is a neurodegenerative disease with a kind of a related disease in humans called Creutzfeldt-Jakob disease. So all these diseases are caused by a misfolded protein. Now this protein normally has a normal function in neurological tissue. So what happens here is that a normal protein, PRPC here on the left, for some reason becomes misfolded and adopts this prion form, and so this is called PRP scrapie. And so this is the abnormally folded form. So we go from these 2 alpha helices, or 4 alpha helices, I guess, and these 2 in the front misfold, and we end up with beta sheets instead, 2 antiparallel beta sheets. These proteins then are able to drive the misfolding of more of the normal shaped proteins. So we have a few normal shaped proteins which become the scrapie form of the prion protein, and then these prion proteins in the scrapie form can bind with the normal protein. That's what's happening over here on the right, and then the misfolded protein causes the normal protein to refold, and so it's kind of like once you get this going, you lead to these large aggregates of misfolded kind of proteins stuck together, and they tend to then accumulate, and you lose the function of the normal protein, and then the misfolded proteins have some adverse effects on the cells. Now, the problem with these is you can't kill these by cooking. So, for example, this is a problem with cows, with mad cow disease, is if people are consuming that meat, there was concern that you could end up with human populations with the same symptoms as mad cow disease. In the US, there's a disease found in elk, a wasting disease called chronic wasting disease, and so hunters had to be very careful about the elk they were catching, and if you know people who hunt elk, it has to be tested, or it used to have to be tested for chronic wasting disease, because you can't make these things uninfectious by boiling them or cooking them or chemically treating them. So those are prion diseases. They're neurodegenerative diseases caused by this misfolded protein, which catalyzes the misfolding of the normal protein to produce these aggregates and accumulations of the misfolded proteins. So that brings us to the end of chapter 4. It's the end of this first set of material, and we'll start on this next set next week. I'll see you then.